Progress.
Okay. Hey, everybody. I'm Terri Conneran with KRAS Kickers, and I am delighted to have you here with us today for another expert series section. Today, we have with us from MD Anderson. I'm Dr. David S. Hong, and he is from the University of Texas MD Anderson Cancer Center, professor, deputy chairman of the Department of Investigational Cancer Therapeutics, phase 1 program. CMD of the Clinical Translational Research Center and the Associate Vice President of Clinical Research. We are beside ourselves to have you with us today because this is really what the whole RAS space is really all about phase 1 and getting us to the next step. So thank you for joining us today. Let me give a real quick shout out that if you are in the witness protection program, if you do not want to be seen on camera, if there's any reason, please change your name, change your picture, and turn off your camera because this is being recorded and we will be putting it on our YouTube channel later. So if you're on, that is giving us permission to share. So welcome with us today, Dr. Hong, and welcome to Amgen for allowing us to do this programming.
Awesome. Terri, for inviting me, kind of last minute fill in for Dr. Marcelo. Gonna try to give you guys an overview relatively quickly. I have a lot of slides, but I do them in about 30 minutes, and then give you guys a chance through Terri to ask questions about kind of next. I apologize, but I do have up at 12 o'clock, and but I will try to do as best as I can to get all your questions answered. Okay. Let me. You guys see my slides? Yeah. Yes,
we can see our slides.
Okay,
and
great.
There's a little bit of an echo, so I can't figure out where it's coming from. So just talk a little slower than usual.
Okay, so these are my disclosures. I'm gonna share this video.
Lift off. Vehicles pitching downrange.
Stage 1 props nominal. Going entry startup. Stage 1 transonic. Stage 1 landing range startup. Stage 1 landing leg deploying.
So the reason I show that is no matter what you think about Elon Musk's politics, he did something that had not been done for 40 years. He transformed rocket technology. He and SpaceX transferred technology since Saturn V. It really wasn't until 2016 that really rocket technology was transformed. And likewise, look at what is happening in the KRAS space. One could argue Dr. Shokat is the Elon Musk of drug development, and it took us 40 years from the time that we actually discovered the pathicity of KRAS to then Shokat developing the first inhibitor that actually worked in KRAS mutant patients. Why was it so difficult? I'm not going to go into too much detail, but there was just a lot of different things of KRAS. It's been hard. If you look at the actual molecule, there's a very smooth surface. There's really limited binding. There's really specifically the ETP or kind of the engine pocket KRAS is very hard to target because of its high affinity, and then other membrane targeted agents like farnesyltransferase inhibitors have really not. Likewise, downstream molecules that signal after has also really not been something that has worked in the last several years. And so it was this ability of Dr. Shokat to develop this molecule that's kind of binds into what is called the switch to pocket near the G2PD pocket. Allosteric just means near. It doesn't mean in the actual pocket. But what he was able to do, design a molecule that not only bound near to the pocket, but in a covalent way, almost irreversible way.
Hang on one second, if you could, David. Let me see if we can get this echo to go away. Can you guys? I'm not sure who's got the main controls for the meeting. Just shut off like everybody except the speakers. Maybe that'll help get rid of the background noise. Let's give it a shot again because what you have to say is interesting and it's a little hard to follow. So with echo.
Now? Okay. So this it locks KRAS into this inactive state. Remember, RAS signaling molecules is called an on and off state. So move on to the next. Okay, so I'm going to go over prevalence of G12C, what the current clinical landscape looks, whether this can be beyond lung cancer. And finally, I'm going to talk a little bit about what are the next steps in KRAS, which I know many of you are in. So some of you guys know this in detail, but these mutations exist really, or RAS exists in really isoforms, K being most common, and within that there are what are called codons, 12 being the most common in the gene, and then within non-small cell lung cancer specifically, G12C is the most common mutagenic allele. But if you look at KRAS, G12C, it exists beyond just lung. Probably the common sub of cancers beyond lung is colorectal. There are other types such as ampullary carcinoma, which is very related to colorectal, solid cancer also. But there are a number of other types, including pancreatic and other primarily gut tumors that also highly express G12C. We looked at this in our group, and we saw that these matched up with the data set in ACR-Gini and other places. And the most common in non-small cell lung, but also colorectal, a bunch of other types. One of the interesting things as we are learning more about KRAS and KRAS G12C in general is that they have also co-operations, other mutations that may ultimately affect whether or not G12C work, including STK11, and this we will look at, and what's called, it's close to it is something called KEAP1, and then colorectal is something called kinase A, which is also important when we think about combinations. This is the paper that I was honored to lead. This was our phase 1 New England Journal paper that shows G12C activity in solid tumors. Typical what's called the phase 1 trial, and then we did in a number of different tumor types, particularly non-small cell lung, colorectal, and tumor types. And it was a relatively well-rated drug. I don't know if some of you guys actually are on this trial. This molecule, sotorasib, the most being diarrhea, some fatigue. One of the things that we are paying close attention to, because this drug can interact with other drugs, increase in the A and ALT. Overall, in non-small cell lung, as a single agent, all dose levels. Remember these are even at various dose levels. We had a partial response of about 30%. In colorectal, however, it was lower. It was about 7%. I'll talk a little bit about what that. In other tumor types, it was around 14. And the waterfall graph. Some of you may know about this. The lower you go, the more the tumor. And you can see here, there were a number of patients who had significant benefit. It's called progression-free survival. How long, in a way, from the time that they started until they progress on the study, we find these through what's called Kaplan-Meier curves, was about 6.3 months. Now, this led to a trial led by my colleague Dr. Skoulides. At some point, you guys will have him come and he'll talk a little bit more about this in detail in lung. But interesting, this led to eventually approval of sotorasib in patients with KRAS G, and showed very as we showed in our phase 1 trial, with even higher response rates of close to 37, and then a PFS of 6.9 months, 6.8 months, and overall survival of about 12.5. So one of the things that we're starting to learn a little bit about the context of KRAS G12C is that there are other mutations that ultimately affect whether or not G12C patients will respond. And particularly, I think this is interesting, is both STK11 and something called, and in patients with KEAP specifically, these patients tend to have worse outcomes than with other mutations. And so if you have KEAP1 and KRAS G12C, that may say that you will probably have less benefit than on KRAS G12C and G12C inhibitors. And that's something that we've generated enough data that Dr. Skolidis may be able to speak on as we move forward. Okay, you may have heard of the other compound adagrasib, which is really closely behind and will, I think, get approval sometime this year in the same setting as sotorasib. And this drug, in many ways, is similar, different than AMG sotorasib. One of the things is the longer half-life. It doesn't necessarily mean it's, but it just means that it exists in blood for a longer period of time. If you talk to Dr. Cannon, who invented AMG 510, say it really doesn't matter because these are irreversible, covalently bound inhibitors, and the trait of KRAS in itself is about 20. So as long as you dose it once a day, it doesn't matter. But my guess is this is far more complex as to how these molecules are kind of processed in body than in animal models. So this was the trial that they led. Dr. Passiani, I'm sure, is another doc you're going to probably have come and speak at some point. Showed a high response rates, 48%, partial response close to 40%. They did have a number overall. You can see this waterfall looks beautiful, and this what are called swimmer plots look also very impressive. And then, but the overall PFS was very similar to sotorasib, and the mean duration response was slightly lower than actually sotorasib taunts. And you can see here the PFS was very similar, 6.9 months from about 14.1 months. When you look at the phase data so far that they've reported, everything looks very similar except for the response rates looks a little bit higher. Time to response, duration response, PFS, overall sort of all look very, very similar, even though there may be some slight differences within the statistical setting. It's very, very similar. Now, there have been a number of other kind of RAS inhibitors, and this is an old slide. I had to update it because there've been a number. There's now, I don't know, a good 15 G12C inhibitors out there. And they're some very good, the Genentech molecule, etc. And I'm into too much detail, but there is many molecules. My guess is this space is going to be very similar to what we see with the checkpoint inhibitors, right? There's like 10 different checkpoint inhibitors out there, all trying to vie for those very, very large non-small cell lung cancer population. Okay, so this is, you may have heard of this, is the Novartis model, which also showed high response rates of 40%. And this was a company from China, also showing high response rate here, close to 43%. Okay, there are a lot of clinical questions, and so we've now answered. One major question is, can this work across tumor types other than non-small cell lung cancer? The answer to that question, I think it's going to be a little to get this approved as a single in all other tumor types. So with agnostic therapy, I wrote a review with my fellows, and there are a number of drugs that are currently approved as tumor agnostic therapy, like larotrectinib, which I help lead. But I think it's going to be difficult for reasons. One is that as a single agent, see here the response rates are much lower. But we do think, this is with adagrasib, we do think that likely in combination, you're going to see increased activity with these drugs. So, for example, this is a very similar story that we saw BRAF inhibitors. When you combine it with EGFR, you can block this feedback loop and lead to higher response rates because you may know, sotorasib as a single agent has very little effect in colorectal cancer. And likewise, we're seeing this pre with sotorasib. We combine it with EGFR, you prevent this kind of feedback loop. And we're now seeing these high response again, 43%, and this was presented last year and was updated recently here at ESMO, showing close to 46% response rates.
Okay, let me just take it. The echo has gotten worse, and I think it might be just a bad signal. Do you think you could call in or try headphone and see?
Yeah,
if
let me
solves
see
it?
if I can.
Yeah, I mean,
Hold on.
yeah,
Let me look at settings here.
yeah, or just I think if Shannon or Caroline can send it to you, and then we'll see if it makes a difference. Because when he's off, am I echoing? I don't hear an echo. So I'm hoping it's just he's got a bad signal or something. So
Is this better, Terry?
yes.
It is better. Okay.
Yes.
Okay.
Yes.
Let me go back to
Okay, we've
sharing.
identified the problem.
Okay. Okay. Let's see.
Houston, we have a problem.
Okay. Is that better?
Oh yes, definitely better. Okay, yeah. Yeah.
I
Because
think
you were stepping on
maybe my
yourself, and so we
computer.
couldn't understand what you were saying.
Okay.
Yeah.
So
Okay, perfect.
okay. So this is. Did you guys? I'll go back here. So this is when we combine it with EGFR inhibitors. You can see here we're seeing this with sotorasib. We're seeing better response rates. And this is likely also going to lead to an approval in colorectal cancer. And this is, and then one other question is, what other tumor types? Well, we're seeing now also activity in pancreatic cancer. Albeit in pancreatic cancer, G12C is probably only present in about 1 to probably 3% of all pancreatic cancers. But in a disease as bad as pancreatic cancer, anything is helpful, right? And so you can see here from our initial phase 1 trial that there are a lot of different tumor types, even melanoma, endometrial, other tumor types that if you have the G12C may actually help and benefit. Okay, I'm not going to go too much into this, but we now know that their patients do become resistant, right? And so we knew this from the get-go that with all targeted therapies, people eventually become resistant. So they've been trying to, Amgen and Mirati and a number of us have been trying to develop combinations. What combinations may overcome some of this and or even work better in patients who have not received the G12C inhibitor. And so this is what's called the CodeBreak 101 study. There have been a couple of other arms that have been added since then. And we're trying to combine sotorasib inhibitors to see whether or not there's any benefit. And so one of the ones that we presented last year was this afatinib, which is an EGFR inhibitor in sotorasib. It was a relatively small study with 33 patients. Unfortunately, one of the problems, and this is something that it's oftentimes hard to model preclinically, is the toxicity. And what we found with this trial was that it's pretty toxic. In combination with sotorasib and afatinib, we saw high grade 3, what are called toxicities, mostly severe rash, diarrhea, et cetera, that made this very difficult to combine with sotorasib. And in the end, what was difficult was also kind of unfortunate is that we really didn't see much higher response rates than single agent sotorasib alone. Likewise, we did this with what's called trametinib, which is a MEK inhibitor, which inhibits downstream. And also relatively small study, total 41 patients, both colorectal, other solid tumor types. And again, we saw high CR rates in combination with trametinib, even at very low doses of trametinib. And again, diarrhea, et cetera, were some of the more common side effects. And we didn't see really that much more activity in combination because, trametinib, I think, can be dosed as high as 4 milligrams. We just weren't able to combine it high enough in order to see if there was increased activity. So again, these are the ongoing trials and arms, and some of these will be reported. In fact, I didn't put this here, but we did see some promising activity with SHP2, and that's been a little bit more tolerable. But several been difficult to combine, particularly with PD-1 inhibitors, these checkpoint inhibitors that recently was reported by Dr. Bob. Okay, so I don't know, does anybody know this date, May 6, 1954? 54? Okay, so this was the day that Roger Bannister broke the 4-minute mile, okay? And the reason I bring this up is this. So remember, before Roger Bannister actually broke the 4-minute mile, there were many, quote, scientific experts who said this was impossible for humans to do, right? And then he did it. He broke the 4-minute mile, and soon afterwards, his buddy from Australia, I forget his name, even got a faster time than Roger Bannister. It's almost like it was a psychological barrier. And likewise, I think in drug development for a long time, we did not well, see, really could, be targeted. They, it was one of the big undruggable targets. Well, that's led to now new targets. And this is totally broken open. KRAS G12D inhibitors, which are the ones that are the only one. There are other companies that Araska and others, and that are likely going to come down, the policy D inhibitors. And this is important because the most common mutation in pancreatic cancer is KRAS G12D, right? There are now new companies like this. You may have heard of this Revolution Medicine company that is targeting all, not just G12D or C or V, but all of RAS. And this could target both KRAS and NRAS. And there are these RAS-on inhibitors. And I'm not going to go into too detail about how it works, but this trial is actually active and ongoing right now, not only here, but about 15, 20 sites throughout the country. So this is one of the hottest studies. Try to get on the study. We don't have, there's not enough data right now. It's still early in the start of this trial. To that, we're enrolling, but this is a trial that everybody's anxious to see if this thing will work. That I think is, I know Marcelo was going to present some of his initial data, is this idea of immunotherapy in T cells. So it turns out that these mutations can be expressed on cancer cells of what are called not just KRAS, but other things like P53, other mutations. And we now can develop these what are called TCR, CAR TCR cells, or T cells where we re-engineer them, to antigens with what are called TCR receptors. And this was recently published by Dr. Eric Trans from Dr. Rosenberg's group, where they targeted a KRAS, I think this was a G12V mutation in a pancreatic cancer patient with a re-engineered TCR T cell. And you can see here the response that they received in one patient in this setting. So in my understanding, in this one patient, this patient has remained on study for a long period of time. So this really, speaks to actually develop, cellular therapies that may even develop, it's called adaptive immunity. Immune system to target KRAS. Okay, so I wanted to, so that's essentially the end of my slides. In conclusion, G12C inhibitors we know are active, particularly non-small cell lung cancer. There is a differential response across ologies, but I don't think it's likely going to be tumor agnostic. The combinations remain a challenge and little improved efficacy, but I do think that we will see some combinations down the line that may be of benefit. And there are a lot of new emerging new therapies in KRAS, particularly immunotherapies and G12D on and crack open all of RAS. This is our team, our Astros team here in Houston, our group that update this. And these are all the people who have been helping. And of course, our patients, you all who are involved in clinical trials, we are incredibly grateful. Okay. All right. I have questions. Let me try to answer as many questions as I can, because I got, see, I told you, 35 minutes for you.
You did. You rocked through it. All right, so I know that I saw something in the chat, so I'm gonna circle back to it, but I know recently I saw Bridge Bio, and I can't remember the name of the drug. It got like a fast track sort of a designation for in conjunction with, I believe it was. with
Yeah,
the
it's
G12
a
inhibitor.
SHP2 inhibitor. It's
Yeah.
from Navier. It's from Navier. Navier. We actually, MD actually developed the molecule here, and I'm running that trial in combination with sotorasib. I think that we're still doing the we haven't gotten that amendment activated at this point. A combination of SHP2 plus a KRAS inhibitor, right? G12C inhibitor particularly. The reason is that what SHP2 does is this. So KRAS is kind of turned on by SOS. It's SOS, and SHP2 kind of helps SOS bridge that on state of RAS into kind of like it's like a best way to say it is like kind of a a truck that helps move that on RAS complex to also BRAF to activate that whole cascade. SHP2 inhibitor is interesting enough as alone toxicities, and initially nobody thought that we could combine this with KRAS. It's the one that sotorasib has combined with med RMD something's been has been okay. We presented this at the World Lung. We saw increase, at least in those patients who had not received G12C, over 50% response rate in the non-small cell lung cancer patients. So I think Amgen is hopeful. Study is hopeful that combination will eventually lead to even more efficacy of G12C inhibitors. And theoretically, if you think about it, is involved in not just G12C on state, but G12D and V and all these. So my guess is that if the G12D or the pan-RAS inhibitors look like they're active, they're going to start combining.
Okay, so all right, I'm going to circle back. It kind of rolled into the question with the GDC 6036 from Genentech. Okay, so is there a difference as far as how the SHP2 inhibitors are working in conjunction with GI cancers, whether it's colorectal or other GI cancers?
Yeah, we don't have enough on the SHP2 with GI tumors. It's interesting that SHP2 inhibitors, at least Revolution Med is the one who's probably presented the most data. They seem to have activity in other mutations other than like NF1, also involved in turning off KRAS. Patients who have NF1 mutation may have difficulty turning off. They also have activity, we think, in what's called BRAF class 3. We do see patients with colorectal cancer who have these mutations in NF1 and BRAF class 3. I don't know, that may be in BRAF class 3. I don't know, that may be what you're into. But there's not enough data to say they work somehow differently in colorectal. But we are trying to see if these, like with cetuximab, the EGFR inhibitors may have differential effects. If that answers your question.
Well, I think part of it is that the data is just not there to support.
Yeah, the data is not there. There's not a whole lot. Yeah.
Yeah, and so if you're in a GI situation, if you have a GI cancer and a G12C, you're already in a more rare space. How would you make a determination which way you would go? I can hear somebody else talking.
Yeah, if you have a KRAS G12C and mutation in colorectal, for example, how to get on to one of those trials, the Crystal 10. I think Amgen is starting their phase 3 trial where combine sotorasib with their EGFR inhibitor panitumumab. So that's best likelihood. We do think that there's going to be activity with chemotherapy plus usually in colon cancer, for example, you usually give FOLFIRI plus irinotecan and cetuximab, or FOLFIRI and irinotecan, FOLFIRI plus PEM. And it's not clear right now whether or not that's going to be safe. We, for example, in the CoBreak 1 arm, where we're combining irinotecan, panitumumab, and we think that likely that's going to have, if not equal efficacy, probably better efficacy than just STAB plus or panitumumab plus sotorasib. So if you are a colorectal cancer G12C and have not seen a G12C inhibitor, I would try to get into one of these trials. And we have this trial open here, or there are several sites in the country that are open with the CoBreak.
Well, and we do have an international audience too, so
Yeah.
it's not always available to every place around the world as yet. So how would you decide if you would use I don't know, how would you decide if you went on to a clinical trial? I can hear I'm echoing too. I'm not sure, how would you decide if you go on a clinical trial compared to if you went to using like just sotorasib as kind of therapy? Okay, I can hear myself echoing, so I think I'm the problem.
Am I echoing again? Okay, so how do you decide if you can get single agent sotorasib if you're non-small cell lung cancer? Remember right now, it's only approved in non-small cell lung, and sotorasib is really the small cell lung. Adagrasib may likely get approval, but right now it's only sotorasib. And that's a really complex and difficult question. But if there is a clinical trial center near you and you can get in, well, I encourage that, one, because but helping the wide community in that context. Obviously, you want to be in a setting where you trust the doctor and the oncologist, et cetera. A lot of patients live in very far places, and trying to get back and forth to these clinical trial centers can be very difficult and costly. And so it's a decision you have to make. We know that patients benefit from sotorasib now, and response PFS and overall survival. So if a trial center, then I would guess as a standard of care.
Okay, all right, that sounds good. And all right, I know, I'm scrolling back here. And again, I can hear myself echoing, so I don't know what the problem is we've got today. I know that we were planning on talking about the TCR. Well, are you up to speed on it? And like, you can kind of like elaborate on it. When we went to the RAS symposium, they were talking about the HLA and how that lands out. And so I was surprised to find out that I'm not sure who they are, but they'll, I think it was NIH, will send you a kit to test you to see if you have that to even begin. Can you kind of go through this?
Yeah. So let me talk a little bit about HLA, talk about TCR, and so HLA is kind of, I don't know how to best describe it, but it's just kind of a body that that God created that essentially can take endogenous proteins, or that's what that means, inside the cell, kind of break them up and then express them on the cell surface of any cell, cancer cell, whatever. And essentially that's what happens in the cancer cells. What takes up these mutational proteins, KRAS proteins, the HLA receptor, HLA complex, and takes part of that G12C to the surface of the KRAS cancer cell, and what happens is that you can then target that with something called a TCR receptor. It has to recognize the mutation. Okay, it has to recognize this. Everybody, all of us have a different HLA complex. And usually, and why this is the case, you can ask God if you ever, about this, but differentiated by race. For example, HLA, which is probably one of the more common ones, is mostly prevalent in Caucasians. It's in Asians like me, it's probably less than 5%. And each of these HLAs can be antigens. So with KRAS, as I recall, most common HLA that presents the KRAS allele is 8 and I think it's 4, and or 8 and 1. I think maybe 8 and 1. And prevalence in Caucasians is actually fairly low. Yeah, it's fairly low. It's something like 10. It's more common in Asians. It's more common in Asian phenotype reason. And so I think one of the challenges is with these DR-based therapies is down the line is, what patient populations are going to be eligible for them, right? Obviously here in the United States, it's a mix, right? It's Caucasians, African American, but primarily Caucasians may be eligible for this trial, but they may not be eligible. They may not be eligible just because of the HLA, even though they may have KRAS G12V mutations, et cetera. And not to say that there won't be any who could be eligible, but that's, I think, one of the challenges is going to be one of the challenges of this trial. Not only just the mutation, but you'll have to fit the HLA, the mutation for these TCRs to work, if it'll work.
Okay, and so it's going to become important to look at these co-mutations and whether it's the HLA or something else as the future kind of like starts unfolding.
Yeah, and I know I don't know if you have Dr. Skoulides coming and speaking to you, but he'll share all we're developing probably on like a KRAS, and there's definitely something about keep one that is kind of limiting the inhibitors, right? We've really not had what's called NGS for next just 5, 6, 7 years, relatively widespread, right? And as we get more and more data, we're learning more and more about these co-alterations that may affect whether or not these therapies will work. And there may be other genes that may show that patients even do better with certain co-mutations, but right now, KRAS is probably the biggest mutation that may affect how G12C inhibitors, and we don't know yet whether it'll affect G12D inhibitors because we don't have a G12D inhibitor in the pipeline. We don't know if it's going to affect any of the pan-RAS on inhibitors. So that's something that we definitely are looking. I myself, Dr. folks here at MD Anderson, MSKCC, and recently KRAS4 to really kind of dig deep to understand what's going on in these models and in patients who have these co-alterations, but also can we find better ways of combining drugs, et cetera. So
Is there a different way?
Fingers crossed. This might. Go ahead, Terri.
Is there a different way that the HLA is detected compared to like the KEAP or the STK11? I mean, is it just another co-mutation, or is it?
No, it's not necessarily co-mutation. You can some of the NGS panels will have the HLA on there. I mean, usually I forget exactly. They usually you can test. Like HLA is something like a test that it's a genetic test, but they get it in bone marrow transplant. They get it in bone marrow transplant patients. right? So you have to, when you match your bone marrow transplant patients, et cetera, with their donor, et cetera. So that we've been getting for a very long time in patients with, in transplant. It's just recently, as the cell therapy merged onto solid tumors, that we've been getting HLA. So you can get it. We've been getting HLA. So you can get it. People can get it just by a blood draw. Else here we get a blood draw, or even like you said, they can send you out a kit, and you can get like a cheek swab, a little bit of your the epithelium on your cheek, and then they can send it in your cheek, and then they can send it, assess it that way also. But it's a way of testing the HLA genes.
But it's not necessarily put on your initial genomic
I
report.
don't necessarily think all vendors have the HLA. I think, as I recall, I don't think that like the Foundation One has HLA on there. I think KRAS may have it, Tempest may have it, but I don't think Foundation, at least the DNA Foundation One has it, as I recall. Don't quote me on that, but as I recall, it doesn't.
Well, and they can change what they start reporting out too. So what might be true
Yeah,
a
at some
month ago
point,
could
at some
change.
point, they may absolutely.
Yeah, okay. And then as far as like, and I'm gonna be real cognizant of the time because I know you only have about 15 minutes and you have a hard stop. So there's that as well. Okay, and so we've kind of gone over a little bit when it came to the colorectal. It doesn't feel like I kind of like finished it when it comes to the immunotherapy.
Okay.
And so if we kind of like tie that up, and then we can kind of like move on to some of the long
Yeah,
and maybe the more rare ones.
Yeah, I see. Why isn't there a combination KRAS inhibitor with immunotherapy? So that's a therapy in general does not work in colorectal cancer. We've tried all these checkpoint inhibitors. They've been looking at that in combination with other things. In general, it doesn't work in colorectal unless you have high, which is a certain phenotype. It's a genetic phenotype that can occur in patients with what's called Lynch syndrome or other and other people without that, but those patients, for whatever, if they have this multiple expression, do very well with checkpoint inhibitors. So that's number one. Number two, it has been, and I'm not saying that this cannot happen down the line. To date, we've really had a difficult time combining with a checkpoint inhibitor. And Dr. Bob Lee, again, a friend of mine and colleague who I'm sure you'll have come and talk, we've just really had a difficulty combining it, primarily because of liver toxicity. There's just been a lot of liver toxicity with the combination. Mirati, is hopefully going to present some data showing whether or not they're have as bad as toxicity. Hopefully it doesn't, because really I think the standard upfront therapy for non-small cell lung cancer is carbo, pem, pemetrexed, and pembrolizumab. But he's hoping that you can combine these G12C inhibitors with checkpoint inhibitor. To date, that's part of the reason. There's two reasons, because in general, in most colorectal cancer patients, it doesn't work. And two, we've really had a lot of difficulty today, at least with sotorasib, in combining that with immuno.
Okay, and anybody, just another shout out. Sorry about the echo, guys. We can't seem to quite figure out what the problem is. All right, and then what about like the more rare types? Well, here, let me ask one of the questions I get asked almost every day. Okay, do we have any idea when that G12D drug is going to be hitting like clinical trial? Can we just get like just a whisper as far of a hint?
Yeah, I talk with Mirati every day. And remember, Mirati's not the only company. There's going to be other companies that are likely. I think Erasca presented at ACR. There's also a whole new class of drugs called Protax. And that's really, really interesting. What it does is typically the protein at their kind of engine, the GDP site, like the Sotoras. But what it does is it tags these proteins like the mutant KRAS protein to be degraded. Those are coming down the pike. In fact, I think Astellas presented their preclinical data on G12D. Likely have also down the line a pan-RAS inhibitor, and will also BI Boehringer Ingelheim is also developing something similar to that. So there's all these new classes of drugs that will emerge in the next year or two. I know that sounds too late. And too for a lot of patients, but my best guess is that the G12D is sometime next year. They will have that trial up and running somewhere. We hear.
Yeah, well, we keep hearing coming soon.
Yeah.
We keep hearing coming soon to a lot of them. Okay,
Yeah.
now what about like some of the more rare, like I see G13 is posted in there. I know we've had Kavancho that spoke with us, like as the G12R was like, there's looking like promising, but we like those more rare types.
Yeah, so theoretically, and this is where these pan-RAS inhibitors can target also rare ones, right? G12, 13. Anybody with like NRAS tends to be rare. HRAS is very rare. Interesting with HRAS, with now likely can respond to what are called with these farnesyltransferase inhibitors. Some of you may know.
Okay, wait a minute. You were really tripped over yourself at the echo. So what was it that you said?
Sorry, can you let me try to. I apologize. I don't know.
No, I don't know either. And it's kind of, you filled in last minute, we moved it an hour and now we get an echo. It's like really.
Maybe my computer, is this better?
It seems better. If you just talk a little slower and hopefully if it is a tripping over sort of thing, it will be less problem.
Okay. So HRAS is also very rare. And we now think that tipifarnib in subsets of tumor types like head and neck may also work in HRAS. Our hope is that the pan-RAS inhibitors will also work across these other rare mutations. And that's why everybody's excited about Revolution Medicine, some of these other pan-RAS inhibitors. There's also another pan-RAS inhibitor from Chugai. It's a Japanese company. And there will be other pan-RAS inhibitors that will come into clinical trials soon. One of the challenges, I'll be honest with you guys, with these early phase 1 clinical trials is that it's hard to get, I have lots of patients on, but there's only so many spots. We get, there are 3 spots in a standard, what's called 3+3 trial design. You get 3 patients per month, and it can usually take 2 months for a dose to clear, right? So 3 patients get onto the trial, it takes about a month and a half with all the data and everything in, and then they reopen it again for another 3 patients. And so it is hard to reserve these slots for, before the Revolution Med's trial started here, I got inundated with people wanting to get on the trial, and we screened them, they appeared eligible, and they went on a wait list because I can't promise them all of these slots. And our first patient here was a screen fail, but I lost the slot because there was 15 sites around the country. It went to another site, and all these 20-some patients that I have on my wait list, unfortunately, they can't wait another 2 months to get onto a trial. So we had to try to let everybody know, and we try to get them on something else. So it is a challenge. We do our best, given the fact that, the science is moving so fast. The promise is, these drugs seem so promising that we push these companies to be opening up more slots for our patients, right? But they also don't want to rush into this. And, we don't know, for example, these are first in human trials. And if we kill somebody on the study because of the drug, right, we don't want to be rushing in and killing more people on this study, right? So that's why we have to be careful and thoughtful about how we treat and design these trials.
It is very difficult to wait.
Yeah. I feel like for cancer patients who have metastatic cancer, their timeline is like compressed, right? So every day is like a month. Every month is like a year, right? Right. And I tell that to our CROs and our IRB that we have to get these trials open, because for each patient, like I said, a day is a month is a year, right? A year is a lifetime. So we really want to try to get these trials activated.
Well, we definitely applaud your efforts, and anything that we can do as individuals, as organizationally, like just, I might be known for kind of getting a little jacked up and excited and enthusiastic about stuff. Just put me in front of the crowd. I mean, like, because it truly, they need to understand that we don't have that sort of flexibility, and, a lifetime is a year, and you completely lost that opportunity if you don't even get into it. But at the same time, I recognize the science needs to be there. We don't want something going sideways, and that would be a real wreck. So, all right, I know we have like 5 minutes left recording. Okay. And let's see, I don't see anything like I'm just scrolling through to make sure I didn't miss anything. If there's like a question real quick, somebody just raise your hand and I'll see. Sotorasib and Avastin, has there been any?
Yeah, so there is an arm on the CodeBreak 101 where we're combining a VEGFR inhibitor with sotorasib plus chemo, actually, like we do in colorectal cancer patients. I think that adagrasib also has one of those arms. Genentech study, the GDC molecule, is also combining theirs because Avastin is their drug with their GDC molecule. We'll see. There's some preclinical justification for the combination. It's not as strong as some of the other combinations, but we'll see if that works. And if it does, that's great.
Okay, all right. And then I'm going to just read the question straight out. What do you think the best option for CRC KRAS inhibitors with SHP2 versus KRAS inhibitors with anti-EGFR?
Yeah, I think the best, if you can get onto a G12C inhibitor plus EGFR, or G12C inhibitor plus EGFR and chemo, that's your best bet.
Okay, and do you think it would make a difference if it was not colorectal, it would be pancreatic, or say like appendix or something? Would it make a difference?
Yeah. I think as a single agent, we see higher response rates as that data that we showed in a recent ASCO plenary. About 22% of patients with pancreatic can respond to sotorasib alone. We don't have a whole lot of data with combinations at this point. EGFR inhibitors in of themselves don't work in pancreatic cancer. So there's not a whole lot of data suggests that the combination will, but we are testing it. We're still testing where it's an all-comer. There is a pancreatic, focus in the sotorasib CodeBreak 101 trial.
Okay.
So if you have pancreatic cancer, I would either try to get single agent or try to get onto a combo.
All right, and then viral versus non-viral for TCRT.
Yeah, that's a really good question. There's ways to engineer the TCR gene, TCR cells with a viral vector or something called CRISPR. You may have heard of CRISPR. I don't think we know what is better at this point. We
probably could spend an hour and a half on just that topic, to be honest with you.
There's a lot of science. There's a lot of nuances right now particularly, in the heme space, lymphomas that are really testing these hypotheses. We're trying to translate some of that in solid tumor. But the solid tumors, we don't have a cellular therapy that is approved, right, in solid tumors yet. And I think we will in the near future, but right now we don't. And so we don't really know what is the best approach per se.
Okay. I think that probably summarizes an awful lot of stuff. Turkey tail, do you have any thoughts on turkey tail?
I have not heard of turkey tail and 5FU. No,
Get out of town, really?
no, I have not heard of that.
Okay.
Maybe that's just, I'm the, is that a certain type of herb?
We'll talk another day. Yes, turkey
Okay.
tail is another type of, it's a mushroom, and a lot of times some like Sloan has a big thing around
Yeah.
it. Some of the integrative stuff does it.
Interesting.
My general shout out on it is I've worked with an integrative oncologist, I've worked with nutritionists, and really check with the team because it can work well with some things and works really poorly with other things. So, don't just think it's the magic cure and it works with everything.
Yeah.
And okay, high dose vitamin C. This does not seem like it's your area of expertise.
Yeah, no, I've heard of high dose vitamin C infusions. There's been a lot of discussions about that, particularly also in patients with sepsis and everything else. But really, I haven't seen a whole lot of data to suggest that it would work in colorectal.
Okay.
If it's there, I just really haven't seen it.
All right, I'm going to shout out these real quick. Metastatic cholangiocarcinoma, G12V, MSI stable, been a trial using nanoliposomal. Okay, have you had 44? Okay, and stayed stable. Okay, that's good news.
That's great.
All right, if there's any, is there time? What do you think about adagrasib for somebody who's had progression with sotorasib?
Yeah, that's a good question. So there have been some reports, and that adagrasib can work in those patients who've been resistant to sotorasib. So some of you, I didn't go into detail about this, but we now know that there are multiple mechanisms of resistance with these G12C inhibitors. I wish it was as easy as just one mutation like in EGFR, predominant one mutation, but it's not. It's like there's a bucket of specific mutations. It's like 15 different mutations, and then the switch to pocket, and then there's another bucket where you have different pathways, and then another bucket even where the tumor changes from adeno to squamous, right? And I wish it was as easy, but I've heard of some reports that adagrasib can still work in patients with sotorasib resistance. And that may just be because of the pharmacokinetics of how that drug is different than sotorasib.
Okay, and then last question, any data regarding appendix cancer with G12C?
Yeah, so remember I said that is probably the second most common appendiceal carcinoma, is common G12C mutant after non-small cell lung. And we did see in the phase 1 study, some patients who did, it's rare, it's incredibly rare, but that we did see responses in patients with G12C as single agent.
All right, it looks like, I think I have a whole minute to spare. Thank you for filling in with
no
for
problem.
us this last minute, and I appreciate it, and I look forward to seeing you, and we'll circle back here real soon. So, all right, everybody else can stay on the community. Thank you so much, David. I appreciate it.
Bye-bye.
Bye.